Namaste students, Kushra Vidya. I, JVWU Dr Chandrakala, Assistant Professor, Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the plant embryogenesis. Plant embryogenesis is a process that occurs after fertilization of an ovules to produce a fully developed plant embryo. This is a stage of the plant life cycle that is followed by the dormancy and germination. Dormancy, that means a period of where the organism or a life cycle when growth, development and physical activity is temporarily stop. And germination, where the process in which the growth of the seed and similar structures that terms applying for the sprouting of a seedling from a seed of an angiosperm or a gymnosperm that goes into a plant. So the zygote produced after fertilization must undergo various cellular divisions and differentiation to become a mature embryo. Zygote in eukaryotic cells form from a fertilization event between the two gametes. Okay. So at the end of the stage of it, which have five major components of shoot, apical meristem, hypopotyle, root meristem, root cap, and the cotyledon. Okay. So unlike the animal embryogenesis, plant embryogenesis also result in immature from the plant starting from the structures like leaf and the stems and the reproductive structure. So the embryogenesis occurs naturally as a result of single and double fertilization of the ovule, giving rise to the two distinct structures. So in a plant embryo, the embryonic or endosperms which goes on a seed into a seedling, the zygote goes through the various cellular differentiation and division in order to produce a mature embryo. These morphogenic events from the basic cellular pattern from the development of the shoot, fruit, or body or primarily division tissue layer, which forming a meristematic tissue which follows the morphogenic events following the particular to the eudicots or and not in monocots. Okay. The two cell stage. So here the two cell stage following the fertilization and the zygote and the embryo are present within the ovules and are seen in first stage of. Okay. So the zygote undergoes some asymmetric transverse cell divisions which give rise to root cells. A small apical cell resting or above a large basic cell. These cells, two cells, are very different and give rise to different structures establishing the polarity in the embryo. Okay. So the apical cells, small apical cells on the top of the content, most of the cytoplasm or the aqueous substances found within the cells from the original zygote. It gives rise to the hypopotyle, shoot apical meristem or the cotyledon. The large amount of the bottom of the content having a large vacuum giving rise to hypophysis and the suspensor. So 8 cell stage at about 2 rounds of the longitudinal divisions and which forms a transverse division. And 8 cell stage is the result. So the second stage in which the illustration about the indicates the which the embryo looks like during the 8 cell stage. According to here the 4 distinct domains during the 8 cell stage is formed, and the second domain, that is the central embryo domain, gives rise to the hypopotyle, and the 16 cell stage which lead to the 16th cell stage, and the 4 domain are still present. So the globular stage, where in this stage is an indicative stage in the embryo presence in the embryogenesis. So the spherical or a globular stage, where the stage three of the stage three has been formed. So the ground meristematic will go on the forming a ground tissue, which includes the pit and the cortex. So the procambium will eventually forming a vascular tissue. So the heart stage, or according to the events here, or finally the starts to form the elongate and the pro-embryo stage, and this stage is defined as the continuous growth of the endocotyledons and the axis elongation. So this is carried out through the entire growth process like any other development, or however in the uh, other different stages or the part of the suspense complex must be terminated, or the suspensor complex is shortened, because at this point of the development, most of the nutrition from the endosperm has been utilized, and there must be some space for the mature embryo. After the suspensor, the complex is gone and the embryo is fully developed. Stage 5 is the where the embryo look like a point of development or maturation, where the second phase or the post-embryonic stage involves the maturation of the cells, and that forms the cell growth and the storage of the macromolecules required for the food and the energy supplies during the germination and seedling growth. So the appearance of the mature embryo is seen in the stage 4. So the dormancy, where the end of the embryogenesis is defined as the arrested developmental phase or a stop or in the growth. So the breaking dormancy or finding a specific requirement of the seeds can be rather difficult here. For example, a seed coat can extremely thick. Okay. So here the role of auxin is the hormone related to the elongation and regulation of the plants, which plays important role in establishment in polarity with that of a plant embryo, and the research which shows that the like your embryonic pattern in the regulation by the auxins and transporting the mechanisms in the polar positioning of the cells within the ovules. So here the inhibitors of this, like during the globular stage of the embryogenesis, the embryos continues the spherical expansion, and in addition, oblong embryos continues the axial. So the somatic embryogenesis is an alternation form of the embryogenesis, where the somatic embryogenesis forms and the somatic embryos are formed from the plant cells that are not normally involved in the development of the embryos. So here the first ordinary tissues or the no embryo or a seed coat is formed around the somatic embryo. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth, Women's University, Jaipur. If you have any queries, we'll discuss in the problem solving.